This conference will now be recorded.
Good morning, everyone. Today's class is on physiology of menstruation. Am I audible?
Yes, ma'am.
Yeah. So this physiology of menstruation we'll just categorize into three headings like hormonal influence, ovarian changes, that is the follicular development and ovarian steroidogenesis, and followed by endometrial changes which are happening due to this hormonal influence. So every month, actually for a menstruating woman, if she has regular cycles, she will have a menstrual cycle every month. So this menstrual cycle will have a ovarian part and a uterine part. See, coming to the ovarian part, the ovarian part consists of folliculogenesis or development of a mature follicle along with ovarian steroidogenesis. This ovarian cycle is further divided into follicular phase and the luteal phase. The first half is the follicular phase where the development of follicle happens up to the dominant follicle followed by ovulation, and after ovulation the phase is called as luteal phase where the granulosa and theca cells convert into luteal cells. Coming to the uterine part of the menstrual cycle, the hormonal influence which is happening on the uterine endometrium will be again divided into proliferative phase and the secretory phase. So this proliferative phase actually coincides with the follicular phase of the ovarian cycle, and the secretory phase coincides with the luteal phase. That is, proliferative phase is under the influence of more of estrogen, and secretory phase is under the influence of progesterones. So follicular phase, the average length of the follicular phase is usually variable. For a woman, if she has a variable length of the cycle, usually the follicular phase will have the variability comparative to the luteal phase, which is actually constant for 14 days. So average length varies from 10 to 14 days, whose variability in this length is responsible for most variation in the total menstrual cycle length. Coming to luteal phase, it occurs after ovulation to onset of menses. Luteal phase average length is 14 days. So luteal phase is a constant phase and follicular phase usually varies, which actually gives the change to the menstrual cycle length. So in a normal menstruating woman, this menstrual cycle is under the control of cyclical hormonal production from the ovaries, that is ovarian steroidogenesis, that is estrogen and progesterone, which parallels the proliferation of the uterine endometrial lining, which prepare the endometrium for either implantation. If implantation doesn't happen, it will shed off as a normal menstruation. So normal menstrual cycle usually lasts from 21 to 35 days, that is 28 plus or minus 7 days, with a flow of 4 plus or minus 2 days, with the average blood flow of 20 to 60 ml. Please remember, normal menstrual cycle lasts for 28 plus or minus 7 with a flow of 4 plus or minus 2 and an average blood loss of 20 to 60 ml per cycle. So each ovarian phase is divided into follicular and luteal phase, and follicular phase usually have a variable duration, and luteal phase remaining constant for an average length of 14 days. And this menstrual cycle variation, length of menstrual cycle variation is usually due to follicular phase. For a normal menstrual cycle, this follicular phase and luteal phase are under the control of cyclical hormonal production from the ovaries, which usually parallels the uterine endometrial lining changes, which prepare the uterus for endometrium if fertilization happens. Prepare implantation if fertilization happens, or else it sheds after the hormonal influence. So this is the menstrual cycle characteristics. You can see the ovarian phase, endometrial phase, and the estrogen phase. The first 1 to 5 days, which we call it as early follicular phase, according to ovarian cycle, and endometrial phase is a menstrual phase. So usually women have a flow of 1 to 5 days, and the next 6 to 14 days, it's a follicular phase in the ovarian cycle and proliferative phase in the endometrial cycle. And the later 15 to 28 days, that is after ovulation until menstrual cycle, start of the bleed. Ovarian phase we'll call it as luteal, and endometrial phase is secretory. And these are the hormones which actually predominate. Usually during early follicular phase, all estrogen and progesterone are in a low level, and in the follicular phase usually estrogen dominates, and the luteal phase usually progesterone dominates. So this is the HPO axis. It should be intact for a normal menstruation to happen. See, from the hypothalamus, the gonadotropin releasing hormone is actually released into the anterior pituitary, which stimulates anterior pituitary gonadotropes to release gonadotropins, that is follicular stimulating hormone and luteinizing hormone. And this follicular stimulating hormone usually it will have more effect on the growing follicles. I mean, it helps in maturation of the follicle or growth of the follicles. And this LH, it will again help for the later stages of maturation of the follicle, and it also helps in the luteinizing after the ovulation. That is, the granulosa and theca cells form corpus luteum after ovulation under the influence of luteinizing hormone. And this growing follicle usually secretes estrogen. Therefore, there is estrogen predominant environment or atmosphere in the follicles. Due to increased estrogen in the mid-cycle, which stimulates LH surge, and with LH surge that happens ovulation. Due to increased estrogen, which have a negative feedback on FSH and LH. And during the later period, that is corpus luteal, luteal phase, when the corpus luteum is formed, there is excess of progesterones, and increased progesterones and estrogen after ovulation usually inhibit the gonadotropins like FSH and LH. So this is a negative feedback. But if you see during ovulation in the mid-cycle, there is increased estrogen which have a positive feedback. You can see this red one, this positive feedback, which also increases LH, which happens where LH surge happens, which follows the ovulation. I will explain you in the later slides. So coming to the embryology in brief, usually ovary has primordial germ cells, granulosa cells, and the ovarian stroma cells. These primordial germ cells are actually from the endoderm of the yolk sac. By 3 weeks of gestation, usually you can see primordial germ cells in the yolk sac, and by 5 to 7 weeks, they will migrate to the hindgut and gonadal ridge. The primordial germ cells from the yolk sac of the hindgut usually migrate to primordial, gonadal ridge by 5 to 7 weeks. And granulosa cells develop from the coelomic epithelium, and the stromal cells from the mesenchymal cells. So after migration to the gonadal ridge, they usually start their mitotic divisions. So they will increase in number. So by 5 months or 20 weeks of gestation, you can see these primordial germ cells increase to 6 to 7 million. At birth, usually we'll have only 1 to 2 million because there is ongoing atresia as well, atresia or apoptosis. These cells usually this cell usually divide, and usually they will some of the cells usually undergo atretic changes as well. Thereby, this number actually reduces. By 5 months, it is 6 to 7 million. At birth, it is only 1 to 2 million. At puberty, it is 4 lakhs. But among these 4 lakh oogonia, only 400 to 500 ovulate. So, in a life cycle, usually a woman will have only 400 to 500 menstrual cycles. So, follicular atresia, which is actually active process due to apoptosis, which is under the hormonal control, starts even if the fetus in utero. Therefore, this atresia or apoptosis process starts in utero as well. So this is the changes of oogonia. You can see the peak or 6 to 7 million at 20 weeks. At birth 1 to 2, puberty 4 lakhs, and only 400 will ovulate. This is the variations in gonadotropes during the life cycle. During the fetal period, you can see there is high FSH and LH in the fetus. At birth, it is reduced because due to the influence of maternal hormones. And maternal hormones have an influence on the fetus. This FSH and LH are at low level. And after birth, again, the ovarian stenogenesis doesn't happen. Therefore, the FSH and LH is at peak again. And before puberty, usually you can see the nighttime increase of hormones. Usually the daytime will be in childhood, day and night, the hormones FSH and LH are in the lower levels. But before puberty, usually there is night increase, nocturnal increase of LH. And after puberty, during the reproductive periods, again both day and night, the LH and FSH are in the mid level, and it will vary according to the menstrual cycle. And following menopause, usually they will peak because there is no influence of estrogens. Because the ovarian, I already told you that ovarian ovulation only happens for 400 to 500 cycles, after which the ovarian follicles are apoptotic or atretic. There are no remaining ovarian follicles, thereby there is no steroidogenesis, no estrogen and progesterone, production. Therefore, there is no negative feedback which is hampering this FSH and LH. So FSH and LH are in the peak level. So briefly we'll discuss about gonadal peptides. Now we are talking, we have been talked about steroidogenesis, that is steroid hormones, that is estrogen and progesterones. Now this gonadal peptides, inhibin, activin and follistatin usually have a control on this cycle. This inhibin usually have inhibitory action on FSH. Activin usually stimulates FSH, and this follistatin, it suppresses the FSH beta gene, and most likely thereby preventing the interaction of activin to this receptor. Therefore, it is inhibin and follistatin also has a negative role, and activin usually stimulates the FSH. During early follicular phase, usually when you see early follicular phase, as we have already said, that menstrual flow, the 1 to 5 days, usually all the hormones are in the lower level. Therefore, inhibin is reduced, estrogen is reduced, and progesterone is reduced, and there is a slight increase of FSH because estrogen is reduced and inhibin is reduced. Inhibin is having an inhibitory action on FSH. When inhibin is at low levels, FSH is at high levels. And FSH is increased, and it will stimulate the growing follicles. And these growing follicles usually secrete inhibin B by granulosa cells and estrogen as well. And this inhibin B, again, it will have a negative feedback on FSH, and FSH levels decreases. Increased inhibin B levels blunt later FSH secretion in the follicular phase. So after which the ovulation happen. For this ovulation, there should be a increased level of estrogen, which will actually have a positive feedback on LH, which cause LH surge followed by ovulation. After ovulation, there is luteinization of this granulosa cells. Granulosa cells and theca cells become functionally specialized cells called as luteinized cells, which will secrete progesterones. And during this progesterone, phase, we will call it as luteal phase. And this inhibin production comes under the control of LH and switches from inhibin B to inhibin A after luteal phase. So first FSH is raised, and with FSH usually stimulates the granulosa cells and follicular development, and inhibin B is secreted by granulosa cells, which have a negative effect on FSH. Thereby FSH is reduced, followed by estrogen surge, followed by LH surge, ovulation, and remaining 14 days of luteal phase. So inhibin B levels peak with LH surge, whereas inhibin A levels peaks a few days later in the mid-luteal phase. I already said after luteal phase, this inhibin B is actually whose action is taken over by inhibin A. So all inhibin levels decline with loss of luteal function and remain low during luteal follicular phase. When the luteal function is at during the menstrual cycle, if the fertilization doesn't happen, this corpus luteum will change into corpus albicans, and there is no production of progesterones and inhibin as well. Therefore, these inhibin levels are at low levels when this corpus luteal function is low during the luteal follicular transition. And this insulin-like growth factors usually mediate ovarian function. Insulin-like growth factor 2 is involved in primordial follicular development. I will discuss in the later slides the follicular development. And insulin-like growth factor 1 and 2 stimulate growth of secondary follicles. 2 for primary follicles, 1 and 2 for the growth of secondary follicles. And gonadotropins usually stimulate insulin-like growth factor 2 production in the theca cells, granulosa, and luteinized granulosa cells. FSH also mediates expression of insulin-like growth factor. All these are at a higher level. You just have a look. That's all. So this is the follicular development stage. It starts from primordial follicle, after which it changes to primary, later on secondary, tertiary, and finally it grows into a Graafian follicle, and finally ovulates, releasing the oocyte. If you can see here, usually the primordial follicle is arrested in meiosis 1, prophase diplotin phase of meiosis 1, and after ovulation, usually the meiosis 1 is completed. Secondary oocyte, meiosis 2 begins and it arrests at metaphase. You can see here, primordial follicle is in a stage of meiosis 1. It is arrested in meiosis 1 prophase. And secondary follicle, usually when it is selected for as a dominant follicle, it completes the meiosis 1 and enters meiosis 2 and gets arrested at metaphase. So this meiosis 2 is completed after fertilization. So coming to follicular development, we have already dealt with hormones. Now we are, I've already said we are actually categorizing this menstrual cycle or studying this menstrual cycle in the 3 headings: the follicular development, the hormonal production, and the endometrial cycle correspondingly. So coming to the follicular development, it is a process which is designed to allow the monthly recruitment of a cohort of follicles, and ultimately it releases a single mature dominant follicle following ovulation every month. This is a definition for a normal menstruating woman. Follicular development, usually it starts with the recruitment of cohort of follicles, that is 4 to 11 follicles usually recruited every month, and ultimately it grows into ultimately all these follicles grow and only one follicle reaches a mature dominant stage, which is ovulated each month. So initial recruitment and growth is not under the control of gonadotropins. Therefore, up to secondary follicle stage, it is gonadotropin independent. So coming to the stages of follicle, it starts with primordial follicle. If you see a primordial follicle usually consists of oocyte, which is arrested in the diplotin phase of meiotic prophase, meiosis 1 in the prophase. It is surrounded, this oocyte is surrounded by a flat, single layer of flat granulosa cells. So primordial follicles consist of an oocyte which is arrested in the meiosis 1 prophase diplotin stage, and it's surrounded by a single layer of flat granulosa cells. So it converts into a primary follicle. It grows into a primary follicle. That is, here the granulosa cells become cuboidal. So this primary follicle consists of an oocyte, again in the stage of meiosis 1 prophase, with the surrounding cuboidal granulosa cells, which are arranged in a pseudo-stratified layer. So during primary follicle, again, there are some products. This acellular zona pellucida is secreted outside the oocyte. Therefore, primary follicle has a oocyte with a covering zona pellucida and a cuboidal granulosa cells which are arranged in pseudo-stratified layer. Primordial follicle will have a flat granulosa cells. Primary follicle will have a oocyte, zona pellucida, and the cuboidal granulosa cells arranged in pseudo-stratified layer. Coming to the secondary or preantral follicle. This follicle includes the final growth of the oocyte and further increase in number of granulosa cells. So this in the secondary or preantral follicle, the granulosa cell number increases and the stroma cells actually differentiates into theca externa and theca interna. You can see here, this is primordial follicle which have a oocyte and a flat granulosa cells. And this oocyte is in the stage of meiosis 1 prophase diplotin. And this is primary follicle, it is having an oocyte and the zona pellucida, which is an acellular layer secretion, and it is covered by a cuboidal granulosa cells arranged in pseudo-stratified layer. Pseudo-stratified means it is actually a single layer, but under microscope you can see it is a multiple layer because of the arrangement of the nuclei. Coming to the secondary follicle, secondary follicle have an oocyte, zona pellucida, and an increase in number of granulosa cells which are arranged in layers, and the theca cells get differentiated into theca interna and theca externa. Coming to the final development of the follicle, that is tertiary follicle. Tertiary follicle or antral follicular, secondary follicular or pre-antral follicular. And this tertiary follicle will have an antral fluid. See, you can see in tertiary follicle, these granulosa cells, among which have a they will develop a spaces filled with fluid and followed by this fluid filled antrum formation. Tertiary or antral follicle. In this, the follicular fluid gets collected between the granulosa cells, producing a fluid-filled space known as antrum. And the granulosa cells are differentiated from this fluid. The cells which are surrounding the antrum are called as mural granulosa. This is the antrum. This is the cavity or the fluid-filled space. And the cells which are covering this are called as mural granulosa cells. And the cells which are covering the oocyte are called as cumulus oophorus. You can see here, this is cumulus oophorus and this is mural granulosa cells. And this is the antral fluid. This is primordial follicle. You can see here oocyte with the flat cells. This is preantral follicle and this is the antral. Preantral is a secondary follicle where granulosa cells increase in number and theca cells differentiate into interna and externa. This is the antral follicle. This is the cumulus oophorus and this is the mural granulosa cells. This is the antral cavity with antral fluid. So how this antral fluid is formed? This antral fluid is actually a plasma filtrate and it is secreted by granulosa cells itself. And this antral fluid is having a high concentration of estrogens. So it is estrogen dominant follicle. Further accumulation of antral fluid results in rapid increase in the size of the follicle and it changes into a Graafian follicle. So early stages of development, as I've said, up to secondary follicle is not under the influence of gonadotropins. Therefore, we call it as gonadotropin independent. And final follicular maturation requires adequate amounts of LH and FSH. Therefore, these are gonadotropin dependent. This is again another picture. This is primary follicle, primordial follicle, primary follicle, secondary or preantral, antral or dominant follicle. After antral follicle, due to this follicular fluid accumulation, the size of the follicle increases and finally it ovulates. So that is the follicular development which happens in each menstrual cycle. Coming to hormonal changes which are happening in the menstrual cycle. These hormones actually we will read it as a two-cell, two-gonadotropin theory, because the two cells are granulosa cells and the theca cells, and the two gonadotropins are FSH and LH. Ovarian estrogen biosynthesis requires the combination action of two gonadotropins on two cell types. So for estrogen synthesis, we need two cells, theca and granulosa, and two gonadotropins, LH and FSH. Here you can see these theca cells. Theca cells are actually have a receptor of LH and granulosa cells will have a receptor of FSH. And if you can see, theca cells will have all the enzymes which can convert for the steroidogenesis. That is, from cholesterol, it can synthesize androgens, androstenedione and testosterone. But if in granulosa cells, these enzymes are absent. So in granulosa cells, the cholesterol cannot be converted into steroid hormones. Therefore, these androgens which are synthesized in the theca cells, they will pass through this basement membrane into the granulosa cells and finally they get aromatized into estrogens. So this is two-cell and two-gonadotropin theory. So this concept is known as two-cell theory of ovarian steroidogenesis. Until the late antral stage of follicular development, LH receptor expression is limited to thecal component. I've already showed you in this figure. LH component is actually restricted to this theca, and FSH is limited to granulosa cells. FSH is limited to granulosa cells. This 17-hydroxylase enzyme, which is required for the conversion of cholesterol to androgens, is present in only theca cells and it is absent in granulosa cells. When this enzyme is absent, the cholesterol is not getting converted into androgens, which are necessary for estrogen synthesis. Therefore, we need theca cells for synthesis of androgens and they are converted into estrogens in the granulosa cells. So granulosa cells are incapable of producing androgen precursors to produce estrogens because it is lacking 17-hydroxylase enzyme. So these androgens which are synthesized in theca cells are diffused into the granulosa cells through the basement membrane. So these androgens, which are synthesized by the enzyme which is present in the theca cells from the cholesterol, they diffuse into the granulosa cells, the basement membrane, and granulosa cells will have a rich aromatase activity. Aromatase is again an enzyme which is required for conversion of androgens to estrogen. So in contrast to theca cells, granulosa have high aromatase activity in response to FSH stimulation, because granulosa cells are under the control of FSH stimulation. So thus these cells efficiently convert androgens to estrogen, and primarily the potent estrogen being the estradiol. So you all understand, right? 17-hydroxylase enzyme is absent in granulosa cells, which is required for the conversion of cholesterol to androgens. So this cholesterol is converted to androgens in the theca cells under the influence of LH, and these androgens, they diffuse through the basement membrane to the granulosa cells, and they are aromatized to the estrogen under the influence of aromatase enzyme with FSH stimulation. So therefore this is two-cell, two-gonadotropin ovarian steroidogenesis theory. So during the end of the previous cycle, usually this estrogen, progesterone, inhibin, all these hormones are decreased with a corresponding increase in FSH. See, inhibin and estrogen are in the low levels, which have an inhibitory action on FSH. When these estrogen and inhibins are at low level, there is corresponding increase in FSH. So this FSH actually stimulates the granulosa cells, and granulosa number increases and aromatase expression increases. It also increases the LH receptors on granulosa cells, and during the late follicular phase they produce progesterone. So towards the end of the follicular phase, increased estradiol levels of up to 200 picogram per ml is required for 50 hours for LH surge to happen. So for LH surge, which is required for ovulation, there is a prior estrogen surge up to 200 picograms per ml for maintained for 50 hours. So this is the hormonal changes which are happening here. This is the follicular phase, that is the development of the follicle. You can see here the estrogen is picking up. Just before ovulation, there is estrogen surge, which is actually maintained for 50 hours for the LH surge to happen. So estrogen should be maintained to 200 picograms per milliliter for 50 hours for this LH surge. So after LH surge, there is ovulation, and after that there is progesterone, increase of progesterone due to luteinization or corpus luteum cells. So the follicular phase consists of the first 14 days, or it is variable, the first half of the cycle starting from the last menstrual period to the ovulation. During the end of the previous cycle, usually there is decrease of hormones, as we have already read. Estrogen, progesterone, inhibin is reduced, and there is corresponding increase in FSH gonadotropin levels because there is no negative feedback. So this increase in FSH is responsible for the recruitment of cohort. So I have already said this follicular development starts with the recruitment of a cohort of follicles that also contains follicle which is destined for ovulation, that is the dominant follicle. So in a woman whose ovarian function is decreasing, usually the FSH levels are elevated. Therefore, the day 3 FSH value is elevated in a women with waning ovarian function. Waning means decreasing ovarian function. So during early follicular phase, there is increase in FSH, which actually recruits the cohort of follicles along with the destined dominant follicle. So during mid follicular phase, during mid follicular phase, again the follicle produces estrogen and inhibin, which results in FSH through negative feedback mechanism. So early follicular phase, usually the hormones estrogen and inhibin are lower level, which increases FSH concentration, and this FSH is responsible for selecting a cohort of follicles. And during the development, and this FSH actually stimulates the or starts the development of the follicles. And the granulosa cells during the development usually produce estrogen and inhibin, which again have a negative feedback and decrease FSH decrease the FSH concentration. This drop of FSH level is believed to contribute to the selection of follicle which is actually destined to ovulate. So the dominant follicle usually have a higher FSH receptors compared to other follicles. So when FSH levels are in low levels, Due to more FSH receptors, only the dominant follicle continues to grow, and the remaining follicles become atretic. Thereby, a dominant follicle is selected, or a follicle is selected as a dominant, which is destined for ovulation. So, early follicular phase, there is increase in FSH, the cohort is recruited, and during development, there is increase in estrogen and inhibin from the growing follicles, which decreases the FSH levels. And during because these FSH levels are reduced, and if one follicle, dominant follicle usually have a more receptors, so with minimum FSH itself, it continues its growth and leaving behind the other follicles as atretic. So effects of FSH, it increases granular cell number, increases the aromatase expression, which is required for estrogen production from androgens, and the presence of estradiol, expression of LH receptors on the granulosa cells. And due to LH receptor expression in the late follicular phase, granulosa cells begin to produce small amount of progesterone as well. So after the development of follicles, that is from primary, primordial, secondary, antral, when the dominant follicle is ready, it will ovulate. So at the end of the follicular phase, there is increased levels of estradiol. This may be due to the positive feedback effect. So usually we'll have a negative feedback because when estrogen is reduced, FSH increases. When FSH is reduced, estrogen increases. So this is the negative feedback mechanism. Usually hormones follows. But during ovulation period, there is a change from this negative to positive feedback, where estrogen levels increases, which will actually increase the LH levels. So it causes LH surge. So for a LH surge to happen, this estradiol concentration should be up to 200 picograms per ml for maintained for 50 hours. So after this estradiol surge, there is LH surge, which acts rapidly on both granulosa and theca cells of the preovulatory follicle, and it terminates the genes involving the follicular expression and turns the gene necessary for ovulation and luteinization. So with this LH surge, after estrogen surge, when estrogen concentrations are increased for 50 hours, there is a positive feedback mechanism which will increase LH, and there is a LH surge which converts the genes from the follicular development to genes for ovulation and luteinization. So in addition to LH surge, initiates the re-entry of oocyte into meiosis as well. So the mean duration of LH surge is 48 hours. So ovulation usually occurs approximately 36 to 40 hours after the onset of LH surge. So this LH surge usually maintains for 48 hours, and usually about 36 hours after the onset of LH surge, ovulation happens. So this is the ovarian stigma. So it follows the ovulation. If you see just prior to ovulation, this Graafian follicle is, usually is seen on the surface of the ovary. So the exact mechanism of expulsion is poorly defined, but it is not due to increase in follicular pressure. But it is due to the presence of proteolytic enzymes like plasmin and collagenase, which will actually thin the follicular, which are actually responsible for the follicular wall thinning. So the pre-ovulatory gonadotropin surge, that is LH surge, it stimulates the expression of plasminogen activator and decreases the plasminogen inhibitor. Thereby it converts the plasminogen into plasmin. And it also has a prostaglandins, usually have a high concentration of prostaglandins prior to ovulation in the follicular fluid, which will stimulate the smooth muscle contraction, and thereby it contributes to ovulation. So ovulation does happen due to increase in proteolytic enzymes like collagenase and plasmin, which will usually makes the follicular wall thin. And due to LH surge, there is expression of plasminogen activator and decrease in plasminogen inhibitor, which increases the plasmin concentration. And also it increases the prostaglandin concentration, which will actually causes contraction of the smooth muscle in the ovary. So before ovulation, there is LH peak, more of LH peak and a little bit of FSH peak, and cause of increased FSH is due to small increase in progesterone, and progesterone synthesis usually begin 36 hours before ovulation. So LH surge, estrogen peak, progesterone synthesis and reception of meiosis 1 do happen during LH surge. And meiosis 1 is completed at ovulation, and it will start meiosis 2, and it is arrested in metaphase stage, and only that is resumed after fertilization. So coming to the luteal phase, after ovulation, we have already read about follicular phase, ovulation, and now luteal phase. So after ovulation, the remaining follicular cells, the granulosa cells and the theca cells, they convert into corpus luteum, which is a yellow body. This process usually requires LH stimulation. After LH stimulation, these cells usually they have a both morphological and functional changes known as luteinization. So granulosa and theca cells proliferate and undergo hypertrophy. More granulosa cells, less theca cells. During corpus luteum formation, the basement membrane between the granulosa and theca cells disintegrates and allows vascularization. Prior to ovulation, the the nutrients to the oocyte is only transferred by diffusion process. But after the luteal phase, there is vascularization which happens. So this capillary invasion usually begins 2 days after ovulation and reaches the center by 4th day. So this increase when there is a capillary invasion, when there is a accessibility to the vascularity, low density lipoproteins are perfused into the theca cells, which is used for the precursor of steroid biosynthesis. As I have already said, theca cells are having enzymes like 17-hydroxylase, which is actually required for steroid biosynthesis. So adequate steroidogenesis depend on serum LH levels, and maximum level of progesterone production are observed in the mid-luteal phase. So ovulation can be safely assumed to have occurred if the progesterone concentration is 3 nanogram per ml by mid-luteal phase, that is on day 21. So estradiol levels drop transiently after LH surge. This decline may explain mid-cycle spotting in some women. After estrogen surge, there is LH surge, after which the estrogen concentration is reduced, where some women usually complains of mid-cycle spotting. So corpus luteum produces large quantities of inhibin A, which decreases circulating FSH levels. So if fertilization occurs and there is a blastocyst formation, and if there is implantation, pregnancy occurs, then it is continued. If it doesn't occur, then corpus luteum usually regresses through a process called as luteolysis. Following luteolysis, again the luteal cells undergo apoptosis and become fibrotic, and now its yellow body is converted into white, where we call it as corpus albicans. So if pregnancy doesn't occur, that is if fertilization doesn't occur, this corpus luteum is converted into corpus albicans. If pregnancy occurs, the HCG which is produced by the early gestation rescues the corpus luteum. So for this reason, usually surgical removal of corpus luteum during pregnancy should be followed by progesterone replacement until 10 weeks of gestation, because after which this progesterone secretion is taken over by the placental function. So before 10 weeks, if you're planning for a laparoscopic or any if you're removing a corpus luteum, then you have to supplement the women with progesterone. So coming to the endometrial changes which are going to happen in a menstrual cycle. So this endometrium is having a basal layer and a functional layer. This basal layer of endometrium usually serves as a reservoir. So after every menstrual cycle, after shedding of this endometrium, this basal layer is usually regenerate into a functional layer. So basal layer serves as a reserve for endometrium and regeneration following menstrual shedding. This functionalis layer is the actual layer which is usually shed out during the menstrual cycle. Functionalis layer also has a superficial stratum compactum and an underlying stratum spongiosum. So stratum compactum is a thin layer of glands, which actually thin layer of cells which have a gland next and dense stroma. The underlying stratum spongiosum usually has a loosely organized stroma and connective tissue. So this endometrial changes that happen in a menstrual cycle is actually divided into proliferative phase and a secretory phase. So immediately after menstruation, usually the endometrial thickness is up to 1 to 2 mm. So after which the proliferative phase starts. This proliferative phase actually coincides with the ovulatory follicular phase. So it corresponds to ovary follicular phase, where the glands become increase in length and increase in number, and cells lining the glandular lumen undergoes pseudostratification, and stroma remains compact. And after ovulation, under the influence of this progesterones, the secretory phase starts, which correlates to ovarian luteal phase. Here, which the glands become increasingly tortuous, and glycogen-rich subnuclear vacuoles appear in the lining of the cell lining glands. So the cells which are lining these glands will have a subnuclear vacuoles which are rich in glycogen, and stroma becomes loose or edematous, and there is increased spiral arteries becoming there is increase in spiral arteries along with increase in number and coiling as well. So in proliferative phase, usually it corresponds to follicular phase, where the glands become tortuous and stroma remains compact. And secretory phase, under the influence of progesterone, there is appearance of subnuclear vacuoles which are rich in glycogen, and stroma becomes edematous and spiral arteries become more tortuous. So if fertilization happens and if blastocyst gets implanted, then the corpus luteum is saved from luteolysis by the HCG, which is produced by the growing embryo. If blastocyst doesn't implant, the corpus luteum does not maintain by the HCG, progesterone levels drops and endometrial glands usually begin to collapse. So if this, if fertilization doesn't happen, so finally at the end of the secretory phase, usually the progesterone level drops when the corpus luteum forms into corpus albicans. So thereby the endometrial glands begins to collapse, and there is more infiltration of leukocytes and monocytes, and there is in the vessels, and the spiral arteries usually constrict, leading to local ischemia, and the lysosomes release proteolytic enzymes that accelerate the tissue degradation. And PGF2 alpha, which have a vasoconstrictor effect, contribute to the arterial vasospasm and also induce the myometrial contraction, which may aid in expelling the endometrial tissue. So by the end of the secretory phase, if the corpus luteum, fertilization doesn't happen, the corpus luteum converts into corpus albicans, and there is a drop in progesterone levels. When there is a drop in progesterone levels, the endometrial glands begin to collapse, and there is infiltration of nucleocytes, leukocytes, and monocytes near the vessels, and the spiral arteries usually constrict. When these arteries constrict, there is local ischemia, and there is release of proteolytic enzymes, and the prostaglandin PGF2 alpha will have a vasoconstrictor effect along with myometrial contraction. Usually it will aid in expelling the endometrium. So the entire endometrial functionalis layer, not the basalis, thought to exfoliate with menstruation, leaving only the basal layer, which is actually necessary for regeneration of the endometrium for the next cycle. Following menstruation, re-epithelialization of the desquamated endometrium from the endometrium basalis is initiated within 2 to 3 days after the onset of menses, and usually it completes within 48 hours. So this is the changes. These are the changes in the endometrium during the menstrual cycle. So this is the proliferative phase, and this is the early secretory phase. You can see the glands become more tortuous here, and this is the menstrual phase. So this is the proliferative, where the glandular cells, the stroma is very compact. You can see here. Here the stroma is loose and edematous, and the glands become more tortuous. You can see here red spiral arteries as well. And this is the menstruating endometrium. Any doubts?